let's go ahead and have Strings walking on in. Come on, Strings, that's your cue. All right, before you guys start, I am going to have the panelists introduce themselves. So go to your starting spot and take a step forward. Take a step forward, like we talked about. Okay, awesome. All right, so we will be starting with our Screams group, but before that, I'm going to ask each of our panelists to introduce ourselves. So I don't know if we want to start here. Awesome.
Hi everyone, my name is Chanel Hasten. I'm the Director of Outreach and Community Relations for the nonprofit Alaka Alliance, which means sea otter in Indigenous language. And my background's in marine biology. I have a master's degree in sustainability education. So I love teaching and educating about the ocean, and really excited to be here for, I think, the third year. Maybe?
Yeah, I think so.
Hi everyone, my name is Chloe Kennedy. I am a wildlife biology intern with the U.S. Fish and Wildlife Service. I am based out of Newport, and I get to spend all my free time driving up and down the coast looking at our seabird colonies we have in Oregon. So it's a lot of fun, and I'm a recent graduate, so I graduated with a major in environmental science and biology.
Hi everybody, my name is Patricia McBride and I work at the Oregon Coast Aquarium as an educator, and I'm excited to be here. I studied life masters in education and zoology ecology and love all this stuff, and excited to hear everything that everyone's going to share.
Awesome. All right, well, thank you very much. All right, so I will give you the cue when you get started.
Hello, we are the Streams group. My name's Elizabeth, this is Rylan, Malcolm, Aridia, Chloe, and Preston. For our 8th grade field-based research project, we have been studying the amount of energy from macros in the drifts of local urban streams in Salem, Oregon. Our group's research question is, how much energy in the form of macros is available to fish, birds, amphibians, and other aquatic organisms in Salem, Oregon? Our group's hypothesis was that we would find a healthy amount of calories in the drifts of each Salem Creek. We also predicted that Mill Creek would have the highest average because of its location in a secluded park. Macroinvertebrates, or macros, are tiny organisms lacking a backbone but are visible to the naked eye. They can be found in both urban and rural creeks and streams, and are indicator species of water quality. In fact, some macroinvertebrates are intolerant of things like pollution, sediment, and runoff, while others flourish in these types of environments. Examples of macroinvertebrates are mayflies, stoneflies, caddisflies, green rockworms, and Hamilton mayflies. Examples of non-insect macros are crayfish, worms, and snails. Many birds, fish, reptiles, amphibians, and mammals rely on macroinvertebrates as a food source. Macros transfer energy from basal food sources, such as plants or algae, to upper trophic levels, including fish, birds, insects, and various aquatic organisms. The more energy available in a creek or a stream, the more organisms that can live and thrive in that habitat. Macros are important to the ecosystem, which is why stream health should be a major priority to stakeholders and community members. Since the creeks we studied are located in or near Salem, Oregon, community members and park goers should strive to reduce pollution and the trampling of grasses, bushes, and shrubs in the riparian zone. Also, the city of Salem and Salem Park should continually strive to avoid the use of fertilizers and pesticides near streams and waterways, especially where runoff can occur.
Cold, fast-flowing, clean water is essential to not only fish and amphibians, but also many plants and animals that make their habitat in or near creeks and streams in rural and urban settings. Native foliage enjoys access to clean water and strengthens riparian zones in the process. It also restores creeks and streams, protects it from sediment and runoff. As a result, healthy riparian zones are full of plants and vegetation such as trees, bushes, and shrubs and grasses, which help hold stream banks in place and also trap sediments and pollutions from entering the water. In contrast, an unhealthy riparian zone is full of dirt, moss, and mud, which easily flows into creeks and streams, making the water cloudy and causing aquatic organisms to leave or even worse, reduce their quality of life. Additionally, human disturbances can negatively impact riparian zones and damage macrohabitats in the process. Things like fertilizers, garbage, and deleterious materials enter urban streams, disrupting the flow of energy that they provide. The difference in riparian zone health is evident by the photo. The one on the left shows a very open, grassy space with few trees nearby. There isn't very much greenery and variety of plants. However, the one on the right has many different kinds of beautiful plants with rocks and fast-paced water. The riparian zone on the right is a nice example of a clean and healthy riparian zone.
Macros, such as mayflies, undergo incomplete metamorphosis as they do not have a pupal stage. This involves passing through multiple life stages: egg, nymph, and adults. On average, mayflies live for around 2 years. Macros can be classified into different categories. First are predators, which eat other macro invertebrates and even small fish. Second are scrapers, which consume algae and other organic matter that grows in solid surfaces, like rocks, logs, and plants. Next are collectors, which feed on small pieces of organic matter. And finally, filterers play a critical role in cleaning and purifying the water. Mill Creek holds a significant historical and ecological importance. It was named after a sawmill in the 19th century. The location we decided to go and do research at is inside David Sawyer Park, which is the location where J-Gems, as a school, goes and helps clean and remove invasive plants. The place we decided to research is circled on the map. The riparian zone for Mill Creek is very healthy. Clark Creek is the second creek we go to. It is named after the park it is mainly located at, yet it also flows into Pringle Creek, the last creek we go to. The site we went to research is marked on the map. Clark Creek is a very narrow stream, yet it's an important stream in Oregon, passing through part of Salem, Oregon, before leading into Pringle Creek. Clark Creek also has a healthy riparian zone. Pringle Creek is the last location we go to out of the three. It is also one of the largest streams in Salem, Oregon, leading to the Willamette River. It is named after Virgil Pringle, who claimed some of the land near it. Pringle Creek also has a healthy riparian zone, making it important to keep it that way. The red circle on the map shows the place in Bush Park that we choose to collect macros in Pringle Creek.
In the field, we use D-nets, buckets, waders, and data sheets to collect our data. Back in the classroom, we use Chromebooks, macro information books, and our data sheets. In the field, we would travel to our first location, which was Mill Creek. Put on our waders and head down to our cross-section. At our cross-section, we would designate the 3 spots, upstream, midstream, and downstream, where we would collect macros and decide who would stand in the creek first. Once we had that arranged, group members would enter the creek and stand in their section for 30 minutes. After the 30 mark, we would switch with partners and remain in the creek for an additional 30 minutes. We collected data for an hour total, and we used D-nets to collect our macros. After spending an hour in the creek collecting macros, we would then fill up 5-gallon buckets and shrink the nets into a large white tray so we could count the amount of macros caught in our designated locations. Once we had our totals, we would add them up and divide by 3 to get our average number for that hour. Once we completed our data at Mill Creek, we proceeded to Clark and Pringle Creek to do the exact same thing. We collected in a total of 6 times at each creek.
During the beginning of our research, we hypothesized that we were going to find the largest amount of caloric energy at Pringle Creek because of the size of the stream and overall depth. Our hypothesis was correct because we ended up finding the most caloric energy in Pringle Creek. Our research collection required us to sample 3 different cross-sections of streams at Mill Creek, Clark Creek, and Pringle Creek in Salem, Oregon. Each collection was done at the same time during the day, but the time of year changed as we sampled in the fall, winter, and spring. Our first location was at Mill Creek in Turner, Oregon. The graph shows the 6 times we collected data and the dates that those occurred. We sampled from November through March, with our largest amount of macros collected on March 21st, finding about 1,128 macros in a 24-hour period. Our lowest amount of macros was collected on February 11th at 488 macros. Clark Creek is the second location that we visited. The largest amount of macros that we collected at Clark Creek was on our most recent trip, March 21st, at about 1,640 macros, and the least amount was collected on February 11th with about 808 macros. Pringle Creek is our final location that we visit on our trips. The largest amount of macros that we collected at Pringle Creek was on November 26th, coming at about 1,432 macros. And we collected the least amount of macros on our most recent trip on March 21st at about 528 macros. As shown on the graph at our first location, Mill Creek, we collected on an average of about 860 macros from all of our trips. At Clark Creek, we found an average of about 1,128 macros, and lastly, at Pringle Creek, we found an average of 1,064 macros. As you can see, Clark Creek was the location where we found the most macros, even though it did not produce the most energy. As shown on the graph, we collected on average more macros at Clark Creek's cross-section, but Pringle Creek was the location that produced the most amount of caloric energy per 24 hours at 10,470 calories. At Mill Creek, there was a total of 9,873 calories in the Driffit's cross-section, and at Clark Creek, a total of 5,550 calories in the cross-section. The reason for Clark Creek having more macros but less caloric energy than Pringle Creek is because of the depth of the Talvig. Again, the Talvig refers to the shallowest part of a stream's cross-section. Since Clark Creek's shallowest part is 3 inches deep and Pringle Creek's shallowest part is 6 inches deep, it impacts calculations. For example, when we multiply the average amount of macros collected at each location by the Talvig, even if there are more macros found at one location, if the Talvig is deeper at another, the total caloric energy at the other location could turn out to be more. Our hypothesis was that we would find a healthy amount of calories in the drifts of each Salem Creek. Our data supports this hypothesis. Each stream produced a decent amount of calories in the form of macros. We also predicted that Mill Creek would have the highest average because of its location in a secluded park. This hypothesis was not supported by our data. Pringle Creek yielded the most amount of energy.
After completing our research, it is apparent that each creek we sampled provided an impressive amount of energy for being in an urban setting. However, we discovered that Pringle Creek had the most energy present based on its caloric total. This means that our cross-section of Pringle Creek had more energy available than in Mill and Clark Creek. At Pringle, we calculated 10,470 calories. To calculate this, we need to know the average mass of a mayfly, or batid, along with its length. Thankfully, with the help of Patrick Edwards, a professor and macroinvertebrate expert at Portland State University, we calculated that the average mass of a mayfly from our macro collection was 0.285 milligrams, with an average length of 4 millimeters. Knowing that the average mass of a mayfly equated to 0.285 milligrams, we multiplied that number by the 24-hour average of macros collected from our 6 trips for each creek. For Pringle Creek, the 24-hour average was 1,064 batids, and by multiplying this number by 0.285 milligrams, we learned that the total mass of macros collected at Pringle Creek in a 24-hour period equals 303.49 milligrams. We have to take the depth of the cross-section into account to provide a more accurate count of milligrams of macroinvertebrates available, so multiplying this amount by the depth of our Talvig, which was 6 inches, the total comes out to 1,820.92 milligrams. Converting this number to grams by dividing it by 1,000, it becomes 1.82 grams. Using the constant 5,750 small calories per gram of batids, as shared by Patrick Edwards,
we discovered that in a 24-hour period, 10,470 calories of batons are available in Pringle Creek's cross-section. So what does all this mean? Again, caloric energy in the drift of a stream or creek in the form of macros is necessary as it supports so many other organisms by providing a food source. Though Pringle Creek provided more energy than Mill Creek and Clark Creek, all streams provided an adequate amount and continually supported a diverse population of wildlife, even in city limits. One limitation we experienced was varying heights of water throughout the school year, which made it difficult to collect macros. In addition, water currents were also difficult to navigate and posed problems getting in and out of the water. In fact, on our last trip to Pringle Creek, the water was high and the current was difficult to stand up in, which caused a student to fall in by accident. This resulted in a loss of data and skewed our results, as shown on the graph. A final limitation was obviously human errors in using D-nets rather than the larger triangular nets that Patrick Edwards suggested. Next year's group could build upon our findings by sampling other urban creeks in Salem. In addition, they could sample from a creek in a rural setting or an old-growth forest to compare the results. Doing so would inform them on whether or not Salem's urban streams are providing similar numbers of energy. Special thanks to Mr. and Mrs. Weddle, Mrs. Ames, Mr. Gunter, Miss Izzy, Patrick Edwards, and finally the panelists. Any questions?
So panelists, feel free to jump in there.
Alright, Elizabeth, Rylan, Malcolm, Arabia, is that correct? Yes. Chloe and Preslin, amazing job, congrats on being the first ones up here. That was a great presentation, I really loved all your graphics and your statistical data was really impressive, so bravo to that. I was curious, so, it seemed like February for two of the creeks had a really high number of, organisms collected compared to March was a lot lower. Is there any, do you know anything about the life cycle or, anything like that that maybe could address that for that time of year?
For our findings on kind of in that February area, the water was higher, and in March the water was kind of lower. So that could be part of the reason that we found more macros when the water was higher.
I can do it. I just, wonderful job. It was very interesting. You guys did a great job. You mentioned at the beginning of the presentation why riparian zones are so important. But if you had to sit down with someone who has no idea what a riparian zone is, what would your elevator pitch be to them of like why we should care about them?
A riparian zone is the location around a stream and it keeps sediment and stuff from entering the stream with its plants. It also kind of makes the stream look nicer.
Awesome.
I really enjoyed it. You guys did awesome. Now I have a question. Basically, sorry, I already lost. Oh, I'm sorry. Oh yes. Okay. So you had said all three of the streams like had healthy riparian zones and everything. Had you thought about comparing it to a site that where it wasn't healthy to see if there were like maybe, or did you notice other sections of the stream that didn't look as good like for the riparian area? And you thought maybe for the future, maybe they could also look at like comparing like a not as good area.
We didn't really look at the other parts of the stream so much. We kind of focused on our cross section, but I think it would be a really good idea for future groups to look at other streams.
In the past, also JGEMS has done studies on streams that include a variety. But yeah, we have to focus, right?
Sorry, one other question. So you also mentioned that some of them like indicate pollution more than other ones. Is that something that you guys looked into at all? like the different types? Were they like indicating, or it was like, man, we felt really good about it?
The water qualities were very different at each pond. Like Mill Creek, our first creek, it's more in more of a like nature's part and is less disturbed compared to the other two creeks. Kringle Creek was like the most disturbed with lots of trash from obviously homeless and going over there, like lots of dangerous things. Why we had lower dots on it, but because of the more bigger area and the more water that comes through, we thought it would be more, but
I know that many different macrovertebrates actually like different levels of water quality. Did you look into that at all when you were looking at the stream to see like what the actual macros that you were finding indicated about the water quality itself?
We did not really focus on that. We just counted how many macros or data for the calculations. We did once find a golden stonefly which does not like pollution at all in the Mill Creek, so.
Along the pollution line, did you collect any like drifting pollution in any of your nets or like microplastics?
no, there at Kringle Creek there was a log there that collected a lot, but we tried to avoid that area to get our data correctly.
So this may be like over your heads, but when you were talking about pesticides and stuff near the riparian zone by the city of Salem, do you know if there's any regulation of where they can have pesticides close to the water at all, or anything like that? Did you look into that?
We didn't look into that, but it would be an interesting topic to look into next year.
Yeah, I liked your suggestions for next year about doing it maybe near an old growth forest and kind of having some varying locations. I think that's a great suggestion.
Did you find any macrovertebrates that were particularly surprising or you weren't expecting at all?
I think just the golden stonefly.
We still have like 5-ish minutes for Q&A. Panelists or Mr. Weddle, if you have anything to add.
I just want to tell you one bit of history. We've been at JDEMS working on Kringle Creek for years. There was a chemical spill that wiped out everything. And we started studying the macros right after that. And year after year, the macros came back. And after about 6 years, we actually got a baby coho salmon in the thing. So it meant that salmon were coming back up in the stream that had been cleared out. And one of the ways it got cleared out was by us planting a bunch of stuff in the riparian zone. We planted on all 3 of those creeks. But that's not my real question. My real question is, if you were a fish, which stream would you want to live in?
Definitely no creek.
Mill Creek?
How about the rest of you?
I would say mildew.
Why?
Well, there was more macros found there, but we just found more at Kringle because the depth of our Talbot. So
Anybody else want to add to that?
It was also like less disturbed. Not a lot of people would go there. Maybe some people just walking their dogs, but it's very less disturbed and way more safer.
I would say it was one of the cleaner streams out of the 3, and like Iridia said, not a lot of people come through there as much as the other 2.
It was a really clean stream and it was really nice.
I thought it had like a nice bit of depth.
That also go to Mill Creek because it is more out of the way and one of the cleanest out of the 3.
Yeah, I like Mill Creek. I've always fantasized about getting a little raft and floating down the channel of Mill Creek. Never have. Someday.
We have just a couple more minutes. Were there any other questions before we wrap up our first presentation?
Did you guys, besides the cool Coho salmon, did you come across any other wildlife besides like the mackerel that they would be feeding on while you're out there?
I don't know the name to it, but I saw a small fish here and there at, I'm pretty sure Clark Creek. I don't know the name of it sadly though.
To be clear, the coho was some number of years ago, not this year.
Oh.
That would have made it into the presentation otherwise.
What was the most difficult part of the field data collection process?
I think getting in and out of the water when the levels were high. I would say sometimes the weather was really bad, and I think people would feel a lot less motivated to collect data, and also the water levels that definitely caused us to lose some distance.
And the weather literally leave everybody, like it would get so cold to the point where everybody barely could move their hands, barely move their bodies in general, especially standing in the water for so long. Sometimes we would get wet and we'd have to bring extra clothes because we are very wet. But sometimes some of us fall in it.
No names, no names.
In addition to that, I think the strength of the stream was also a problem.
Yeah, I agree with the stream. In my opinion, it was counting the macros because I cannot really count them, so I got help from others.
Probably like the water levels, they were really like really hard to get out of after you collect that.
Yeah, go ahead.
Are there any things you could do as individuals to help protect riparian zones? Some suggestions you'd give to us here that we try to do locally?
You could pick up litter whenever you see it around riparian zone. Or
I
just
think-
anywhere.
I think also maybe planting variety of plants nearby.
I would also say picking up trash around, because there was definitely a lot of trash that some of the streams, especially.
Okay, awesome, thank you so much. Let's give them one more round of applause.